biology. Uh, Happy Tuesday, day 2 of this online school stuff. And I thought, might as well do a trivia. Just one, because I think it'd be weird to keep asking trivia where no one could answer. But hey, why not one? So, what do you think? Little brown bats of US and Canada, just a common bat species, they can eat up to around how many insects per night? What do you think? 100? No, more. 500? More. Actually, they can eat up to 1,200 insects per night. And lots of those may be things like mosquitoes and other things that we aren't particularly fond of. So, bats are our friends. And a little sad note for bats, there is actually, and this is affecting little brown bats as well as other bats, introduced fungus that they call like white nose fungus that is attacking bats. It's really bad in the Northeast, but it is spreading through the US. And it's called that because their noses literally are covered with a white fungus. It attacks them usually through their winter hibernation, and it will spread from bat to bat through the entire cave and cause up to 90% fatality, sometimes actually even more. It causes them to wake up during hibernation, and then they'll use up their fat reserves and actually kind of starve through hibernation, and they'll go into these caves and in the end of winter, and they're just covered with dead bats. Sad. So I don't know if you guys have been to any caves around here, but I went to the Lewis and Clark Caverns last fall, and one of the things they asked was, have you been to any other caves in the last year? And then if you were wearing any of the same clothes, and they would disinfect those if you had, because what they're trying to do is stop the spread of that fungus and make sure none of it gets in the caves there and affects the bats. So anyway, there's your little FYI trivia. Okay, so a few announcements for today. I know some of you are still needing to get into the school, and today is probably your allotted time to get your notebook. So I'm not going to add anything new as far as responsibilities. However, if you haven't gotten your notebook yet and you get your notebook today, please complete the protein synthesis coloring, which is titled, this was a little confusing, I'm sorry about that, it's titled Transcription and Translation, okay? But it is that coloring that we did before break. And there are 7 questions on the back that I'd like you to complete. I talked about this in one of my classes, but I don't think all of them. There is one new term on the back, so we'll just do that right now. So you can refer back to that when you're doing the questions if you forget, or back to this. But it asks, how many bases are in a codon? And you should definitely know that. It's from your notes as well. But how many bases are in a codon? That's how many mRNA bases code for a specific amino acid. And maybe you remember that, but also it's how many bases you're using on the codon chart. Remember, you have 1 on the left side, 1 up top. You go to where those come together. And then the third base is on the right side of the box. And then that goes over and you can tell what amino acid you're coding for. So there's 3 mRNA bases in a codon. What you might not know is how many bases are in an anticodon, because we haven't used that term before. But an anticodon is just the mRNA bases that the tRNA has that match up with the codon. So if you have AGG for your mRNA codon, that codes for a specific amino acid. Well, the tRNA, how it knows what amino acid to bring is it has an anticodon. And so A would go with U, G with C, G with C, and that would be your anticodon bringing an appropriate amino acid. So the anticodon pairs up with the codon, so it has the same number of bases. So it has 3 bases. So So I hope that helps with the questions. If you have any specific questions on the questions, email me, okay? And then there is the green pedigree sheet. Third period, you are not doing that. But every other class period, you did the green pedigree sheet, and you probably needed to finish the pedigree for the English royal family on the back. That was related to hemophilia, which is a sex-linked trait. And so you can see females can be half shaded in, okay? And males are either blank, meaning they don't have the disease, or they're fully shaded in, meaning they have the disease. Females can be carriers since it's on our X chromosome, okay, and it's a recessive allele. So a female can be X, big H, X, little h, meaning she carries the hemophilia disease and can pass it on to her sons if she passes on the X, little h, because dad will pass on the Y that just makes a male and nothing else. Okay, and so you can look how that has been passed on in the royal family. Side note lesson that we hopefully learn from that and other weird recessive alleles that were passed on in royal families is incest is bad, okay? We don't see those traits as frequently in populations unless there is some incest, and Queen Victoria married her uncle. There was probably other examples as we look back further in that pedigree, because that's how they used to do things in royal families. Gross. So answer those questions. And the last thing I wanted to say today, now from now on out, I'm going to try to give all assignments on Google Docs, things that can be done online and turned in online. However, since this was from before the break, you have a hard copy. Okay. From suggestions of one student and then talking to another teacher who is trying this, we're going to see if you can scan and email those to me. Okay. Maybe some of you have a scanner at home. Nice. But a lot of you don't. That's totally fine. However, a lot of you probably have a smartphone. And there are a few apps that are pretty easy to use. They're free. You just have to use them and then delete what you just used and then do it again, because you can't accumulate files. Then you have to start paying. So it might be a good skill to know. You're probably going to be asked to scan things maybe to a potential employer in the future. So see if you can find a few that I have used that have worked pretty well are Genius Scan. Okay. And I believe it's Tiny Scanner. And then those, you'll just take an image of the front and the back of the paper, and then you can email it to me straight from there. But you'll have to know my address, which is just e_Quintia@cfmtschools.net. All right. Let's try and have them done by Friday and scanned to me. If I don't have them scanned to me by Thursday, I'll probably start calling people to know if you've been watching these videos, and if you got your notebook, and if you figured out how to do that and try to scan it to me. All else fails, you could probably just take some pictures. They're not as good. And they're a lot larger files, but you could take pictures of those and send them to me. Why don't you do one assignment at a time? So do the protein synthesis coloring as one, send it to me, and then the pedigree is another. Don't bother doing the back of the pedigree sheet, because that doesn't ask you any questions. All right, good luck. All this stuff that I've done is new to me. Maybe scanning is new to you, but it's a good skill to have in the future. So give it a shot. Let me know if you're having any difficulties. All right. Have a great day. Bye.